Thanks so much. Good afternoon, everyone. They tell me the first rule of public speaking is to make sure the audience is awake, so I just wanted to test. I know we're late in the day here. But thank you so much for coming. And please, of course, at the conclusion of this talk, don't forget to fill out your feedback. It's right in the Google Cloud Next app. So my name is Jonathan Sheffi. I am our Senior Product Manager for Life Sciences at Google Cloud. Now, over the last couple of days, you've heard about a lot of the amazing technologies at Google Cloud that we're developing that span across the gamut. You've heard about advances at BigQuery, our containerization technologies, compute technologies. We also have a set of teams that are really focused on verticals, focused on advancing specific industries. So today I'm going to tell you a bit about the organization that I'm a part of, the Google Cloud Healthcare and Life Sciences team. Tell you specifically about our genomic data processing solution and what that entails, the set of products and partnerships that make that come to life. And then I've got a couple of special guests. So Anita and Nandi from Oxford BDI, Oxford Big Data Institute, will be joining to talk about her work with the Mosquito Data Atlas. And then Vivek Iyer, who is from the Human Genetics Informatics team at the Wellcome Sanger Institute, will be talking about his work. So at Google Cloud, Healthcare and Life Sciences, we're thinking about a lot of interesting problems because there's so many great data problems in this industry. So not just life sciences, but also clinical data sets. You may have heard a bit if you stopped by the Healthcare and Life Sciences industry booth earlier. You may have seen about our de-identification API, which makes it really easy to strip out PHI from your EMR data or medical imaging data. You may have heard about our healthcare API, which makes it really easy to ingest EMR data from any FHIR or HLV2, try that one more time. Any FHIR or HL7v2 endpoint. And also our DICOM API, focused on medical imaging, makes it really easy to extract metadata from medical images and work with it in Google Cloud, enabling our customers to do analytics on millions of patients. Our team is composed of product managers, such as myself, a large set of software engineers, our solution architects who help our customers bring all this to life and get it deployed, and our commercial team who brings deep domain expertise from the healthcare and life sciences field. Most of us come from the industry, myself included. I myself was originally trained as a computational biologist, spent many years at some of the pharma companies in the industry. So, in life sciences, it turns out the problems don't stop just at the wet lab. The amount of data that we're seeing in the life sciences space is exploding. That's kind of not news to anybody who's come to this talk. Hopefully you've probably heard about with the decreasing cost of whole genome sequencing, the amount of data that's being stored, processed, analyzed, and managed is growing, doubling every year, every 7 months. It creates new problems for us to help solve as Google Cloud. More importantly, the applications are really important when it comes to drug development. How do we make this happen in the real world? Genomics is quickly becoming very much a part of the drug development process. Francis Collins predicted almost 15 years ago that most drugs would become from an understanding of human genomics. We're about up to a third these days. And that's really quite something to know that our understanding of the human genome is already informing therapies that are being approved now. So that brings us to our genomic data processing solution. We see lots of organizations who are generating data off of DNA sequencing instruments, and they need the storage and compute technologies in order to process this data at scale. They need to be able to ingest their data, manage workflows, and control access to the data sets, and be able to run those workflows over and over again on millions of samples. So a great example of this is our collaboration with the Broad Institute, back home, or my home, in Cambridge, Massachusetts. So the Broad Institute currently manages about 60 petabytes on Google Cloud. They process about 600 genomes every day, and have now processed on Google Cloud well over 100,000 whole genomes. And the way that they do this is through our genome processing solution, which includes both products and partnerships. So this is a diagram of the human genome sequencing workflow that many of you may be familiar with. So in order to deal with these defined workflows, which is what's usually used, you may have heard of the Genome Analysis Toolkit, which was pioneered by Broad. You may have heard of Deep Variant. Deep Variant is a machine learning, TensorFlow-based variant caller. So as opposed to using Bayesian inference in order to call variants from mapped reads, instead it treats the read pileups as an image and does machine learning on that image of read pileups. And the accuracy numbers we're seeing from Deep Variant are very exciting. Definitely encourage you to check out the publications. As that data has been processed to BAM and VCF formats, there also leads an opportunity for tertiary analysis in population genomics. This is also an area, I'm not going to talk too much about that today in the interest of time, but we're doing a lot in this space as well. How do you deal with your process genomic data and do analytics on it? We see customers in clinical labs and research settings who are using the tools we've built to ingest their VCF or variant call files into BigQuery. And BigQuery, turns out, is a pretty amazing variant store. And they're joining their variant data to their clinical data, to their phenotypic data, to their medical imaging data, and doing analytics, performing machine learning, and making scientific discoveries using the power of BigQuery. There we go. So some of the benefits here, what makes Google Cloud ideal for genomic data processing? Very flexible pricing. One of the features of our core product, which I'll talk about in a minute, Cloud Life Sciences, is flexible machine types and highly optimized pricing. We make it really easy to spin up a lot of containers. Basically, that's what Cloud Life Sciences does. I'll talk about that more in the next slide. But Cloud Life Sciences, essentially, you go to Cloud Life Sciences and say, I've got 30,000 Docker containers from all the different containerized workflows I'm running. Please run them all. Find the right machine shapes, mount that Docker container and hook it into storage, localize that file, process that file using the relevant step, and return it back to storage. We also provide important security controls, a lot of important workflow engine partners and other technology and service partners, and access to key data sets. I'll talk a little bit more about that in a minute. So some of you may have heard of what used to be called Google Genomics, and Google Genomics is great, but we've actually taken things far beyond that now, and that's why we've rebranded this as Google Cloud Life Sciences and added features. So now Cloud Life Sciences is fully regionalized. What I'm excited to be able to announce on this stage is, whereas Google Genomics had been a globalized service, Cloud Life Sciences is now a regionalized service. So as we moved into beta, which was just happened last month, we launched in Iowa, and this quarter we're launching right here in our London region. So this gives the power for organizations that are based here in the UK to process their data and know that the data, the compute, and the metadata are all staying in the London region. We'll also be launching to other regions in Q1 and Q2 2020. We're really excited about the global rollout here. So the benefits of Cloud Life Sciences for life science organizations, for academic research institutions, the ability to build on open standards and containerized workflows. So some of you may be familiar with open source workflow engines such as Cromwell, Nextflow, and Galaxy. And these have all been made to work for you on top of Cloud Life Sciences. So you're able to use workflow engines that you're familiar with right away. We're also working with proprietary partners, or SaaS partners, who offer researcher-friendly interfaces on top of Cloud Life Sciences and Google Compute Engine. So Terra, 7 Bridges, BC platforms, and many other life science research platforms that you may be familiar with. For researchers, they gain the ability to deal with that data in a friendly format and get to results, get to answers fast. And for IT organizations, they know that they have a solution that's safe, secure, and can scale with their organization. Talked a bit about our partners. I also actually want to talk a little bit more about some of our data sets. So, yeah, I want to talk a little more about some of our data sets. So in 2018, we announced a very important partnership with the National Institutes of Health. There's a program that we announced called the STRIDES program. It stands for something, I can't remember what the acronym stands for, but what it does, we've reached an agreement where we are onboarding the most valuable biomedical data sets into Google Cloud. This includes the Cancer Genome Atlas, the Cancer Imaging Atlas, TopMed, the Sequence Read Archive. We recently have now made available the entire public portion of the Sequence Read Archive, and we will soon be making available the controlled access portion of the Sequence Read Archive. And the important thing is that these data sets are now available to researchers at no cost. So, especially in the controlled access setting, we've also done an important engineering work to integrate the authentication and authorization and workflows you're used to with the NIH. So we know when Professor Jane Doe logs into her Google Cloud account, we are able to associate her identity at Google Cloud with her identity at NIH and soon also at Elixir, and therefore provide access to the right data sets to the right researcher at the right time. So we're changing a flow, what many of you may be familiar with, of filing data access requests, going to the dbGaP portal, fumbling with the credentials and the login, going to an FTP server, downloading that data, finding space in a data center or in a cloud account before I can even get to work, and it adds a lot of delays to the research process. We're changing that flow and replacing it with log into Google Cloud and get to work. We think this provides a lot of important benefits for the research community. Of course, for the researchers, it makes it easier to get started doing your work. It provides a very important benefit for the research computing leaders who support these researchers. I can't tell you how many times I've been in a meeting with a research IT director and a set of researchers, whether at an academic institution or an independent research institution or a commercial institution such as a pharma company, and we find out in the meeting with 3 researchers and the research IT director that there's 10 copies of the 1,000 Genomes Project, 8 copies of the Cancer Genome Atlas, 5 copies of the Cancer Imaging Atlas, and we find out that there's all this multiplicity of data that's being stored in the existing data center or possibly the existing cloud account. And so now we're taking that problem away and saying, well, why don't you just log in, get started on Google Cloud, and get to work. This is just the beginning of the data sets that we're making available. You'll see a lot more of these in 2020. We're investing heavily into this program. So I want to talk a bit about what does this look like in action. We're very excited about this project. So recently, Autism Speaks, which is a major patient advocacy group based back in the States, worked with DNA Stack, one of our Google Cloud technology partners, Sention, another Google Cloud technology partner, and Verily, which is another Alphabet company that is involved in biomedical research, and along with Google Cloud, on a very important new project. They performed alignment and variant calling on over 11,000 whole genomes of people with autism and their family members. They discovered many new important genetic variations associated with autism. And importantly, through the sophisticated use of Google Cloud and our preemptible VM technology, they reduced their cost of processing to less than $3 a sample, which is pretty remarkable. For anyone who's been in the bioinformatics industry for some time, it wasn't that long ago that processing a genome cost $50, $100, and through advances in compute, through sophistication in workflow execution, we've brought that cost down, and we continue to invest in reducing it further, just to enable the scale of research that everyone wants us to see. Importantly, the Missing Project, which is the name of the data set, they even created an interactive data discovery portal for researchers to go and use the data themselves. They've gathered not only the genomic data, but clinical data as well. And as I mentioned, they're using Google BigQuery under the hood in order to power this analytics engine. So researchers can go ahead and get started using this data set and exploring it on their own. We're also seeing a similar project, I'll just mention as a sort of a bonus here, I don't have a slide on this. Anyone who was at ASHG in the American Society of Human Genetics meeting in 2018 may have seen an announcement from Color. Color is a genetic testing lab based in the States focused on cancer. And they had processed tens of thousands, I think around 50, I think it was about 50,000 samples, and said, okay, we've got this all these samples and a good chunk of clinical data from the physicians ordering the reports. We want to understand more and potentially expand the test and make discoveries on this data set. Well, they were able to use BigQuery. They ingested this genomic data into BigQuery alongside their phenotypic data, which was in a Postgres database. And then for their company Hack Week, they stood up a bunch of machine learning tools and analytics tools on top of that joined data set. They actually ended up coming up with a very sophisticated model of clinical disease that outperformed existing clinical models of disease. They also found out that their test predicted a biomarker that has nothing to do with cancer. They won't let me tell you which one it is, but they're very excited about it and maybe publishing that at some point. So we're seeing organizations, both commercial and independent, who are using the power of analytics to make true scientific discoveries. So Thomas Fraser, who's the chief scientific officer for Autism Speaks, I think this says a lot about what we're trying to do as cloud healthcare and life sciences. But with this research, enabling researchers to identify genetic influences on autism that can lead to tailored medical care for those individuals, and really getting to the opportunities for true improvements in treatment and diagnosis is what we're here to accomplish, and it highlights our mission as an organization. So with that, I say thank you so much. And now I'm just the warm-up act. Now you're going to get to hear the people doing the real work, some of the researchers who are using Google Cloud today to do science. It looks like I've got a few minutes left. I'll take a few questions now before I turn things over, and then we can also do some Q&A at the end. I've got about 8 minutes for questions, it seems. It's about where I wanted to land. Stunned, I know. You're so excited. Oh, right here.
So when you shown that number about the processing cost coming below $3 in a preemptive settings, how long roughly does it take to process a whole genome?
It depends. And you mentioned, part of that's because you mentioned preemption, right? And so for those folks who are not familiar with, I just want to explain a little bit what preemptables are, because that affects the answer. So preemptible virtual machines are compute instances that are offered to you at a substantial discount, roughly 80% off, in exchange for the fact that they might be taken away if there's a spike in demand from folks who are using non-preemptible instances. So for this particular project, I don't know the exact turnaround time, the average turnaround time, although I'm happy to find out and follow up. I don't think there's anything confidential about it. What we see often in practice, I've seen for GATK can be on the order of 12 to 24 hours. It's on the longer side, partly because preemption sometimes delays the process. We've also seen some implementations that speed it up quite a bit and get it to on the order of 3 hours or sometimes even less. So I'm not giving you, I think, the exact answer you might like, and the short answer is it depends. But we'd be happy to discuss with you.
These are human genomes, not...
These are human genomes. All we're talking about, today we're talking about human genomics. If you're interested in other species, there's a lot of work happening in plant, rice, other animals. In fact, we're going to hear a bit about some other species in a minute. Yeah. Else? Yes. Hi.
Thanks for the talk. One of the practical problems that I'm currently facing is that data ingestion. The current biological data is all scattered in many research institutes.
Sure.
And, they are quite big. In order to make use of the capability in the Google Cloud, I would have to try to figure out ways to move the data in, or use the data, pipe the data in there, ingest it, and use it as is right there. But I was wondering whether you have any comments on that.
Sure. So I just want to make sure I understand what you're asking. So I think what you're asking is you have data sets that are, say, under the desk of one lab, or under the desk of another lab, or in a sort of a lab server, and migrating that data to Google Cloud? Or are you talking about moving from a data center to Google Cloud, or both, or would?
Sorry, more from the different data centers. Let's say one data center from Sanger Institute, one from another data center, let's say from EBI, and
Sure.
all try to move to the Google Cloud in order to do something meaningful.
Yep. So we have existing transfer appliances that make it really easy to bring some large data sets over. If we're talking about really, really, really big data sets, there are special arrangements that our customer engineering team can work with you on to get those data sets into our data regions. I think you have a follow-up. Go ahead. Yeah, go ahead.
Sorry, one quick follow-up. That Google just made an announcement today about data.
There were a lot of announcements. I can't remember them all.
Sorry, it's about data ingestion, data fusion or data fusion. Yeah.
Okay.
Would there be any data fusion adapters be able to make use of help us that way?
So I don't have a good answer for you. We haven't talked as much about integrating with the data fusion team, but it's a great question. Let me follow up with you. We've exchanged emails, but we can talk about that more separately. Yeah. Yeah.
So with the public data sets, have you curated them in any form, and have you published the curation steps? Or is it as is?
They are provided as is. So I mentioned we have product managers, engineers, and solution architects. I didn't mention that we have scientists. So we don't really have the background with which to necessarily scientifically curate some of these data sets and make judgments on the metadata. So we are not providing that as part of Google Cloud. However, a number of our technology partners, so I mentioned Terra, which is a platform that's developed through a collaboration by the Broad Institute and Verily, does offer some curation tools. And we're also discussing how we can provide more effective metadata management of scientific data sets. Is there a particular use case that you're thinking of?
Any one of them, the usual use case in pharma is that everyone curates them over and over and over and over.
That they do.
Everyone's got.
It's true. Yeah. And I think that if we curated them, to be perfectly direct, I think if we curated them, it would just be yet another curation. And I don't know if we could do it any better than a pharma company could or a scientist could.
It's about having a common source of a common data set that everyone works off, a common curated data set, rather than because each rules might differ. But anyway, that's
if you can get the world to standardize on a common metadata ontology for describing scientific data, I'll give you 20 pounds out of my pocket right now.
I wouldn't take you out on that.
All right. I think I got time. I think maybe for one more, if we've got one. Great. Okay. So next, I'm going to introduce Anita Nandi, who's a research scientist from the Oxford Big Data Institute. She's going to talk a bit about her work with the Mosquito Data Atlas. Anita.
Thanks. Okay, thanks very much. I'm Anita. I'm a researcher at the University of Oxford, and I work on mapping malaria. So just first for a bit of background, as you know, malaria is a disease that's transmitted by mosquitoes. So mosquitoes have a parasite. If they bite a human, they transmit the parasite into the human's blood. If a non-infected mosquito then bites that human, the parasite is then transmitted to the mosquito, and the cycle continues. And this causes a major public health burden globally, causing several 100,000 deaths every year. And most of those are in sub-Saharan Africa. And so there's a global effort to help reduce and eliminate malaria. To do that, we need to know where the malaria burden is. And that's where our research group comes in. We're called the Malaria Atlas Project, or MAP. And we are a very interdisciplinary team of researchers. We're epidemiologists, statisticians, ecologists, physicists, and others. And we provide malaria estimates to estimate the burden, the global burden of malaria. And while malaria is decreasing globally, which is great, This does mean that it becomes harder to model because as burden decreases, we have to develop new techniques to model low burden areas, which is more complex. And so this is something our group does, and we model at very high resolution, very fine scale, because these kind of maps are vital for understanding, for policymakers to understand where to target interventions and how to start eliminating malaria. So we have a lot of malaria data. We have both point data, so surveys conducted across the world on testing people's blood for malaria, so what proportion of people have malaria at this point. But we also have data in regions, so how many people came to hospital with malaria, how many cases are there in this region. And so we deal with large amounts of different types of data across the world. This data can all, by the way, be found on the MAP website and downloaded. And you can see from the map that some places have some types of the data, not others. And so this results in a very interesting and difficult problem of how to model malaria risk across all regions at fine scale. And that's what we work on. And to do that, we use a lot of environmental data. So as we've seen, malaria is transmitted by mosquitoes, so suitable habitats for mosquitoes are more likely to have malaria. So warm tropical climate near stagnant water is at higher chance of malaria risk, things like this. And so it's very useful to input environmental data into our models to give us information about malaria distribution. But not just environmental data, maybe human data, things like accessibility to cities and other such variables. And so these, so we have global satellite images of these variables, and we're mapping at 5 kilometers squared resolution. And so this means in these maps, there's over 18 million pixels in each one, and we use many more variables than this. And so all of this is going into our models. So how do we use Google Cloud? So we're taking all of this data, and what we want to do is we want to run our models on this data to produce global maps of malaria risk. And using Pipelines API, what we can do is we can run a single command telling it where our input data is in cloud storage, what size of machine we want, what container image to use to define the environment, and where to output our results. And with that single command, it then spins up a VM, runs our model, saves the output where we asked, and then shuts down the VM. And so you only pay for that machine for the time your model is running. And so that's very useful for us. But here, in this situation, we can run, it's not too computationally intensive for us to run this kind of model on our on-premise servers, although it is a bit slower. However, this has produced here a single map of malaria risk. But it's not good enough for us to say to policymakers, oh, we think 40% of people have malaria here. They'll want to know, oh, so does that mean between 39 and 41, or could it be 50%? They want to know a level of uncertainty. And these are very important questions when thinking about where to target your interventions. What is your uncertainty? And so to quantify uncertainty, what we do is we run many, in this case, 100 similar models, varying our inputs slightly. And with Pipelines API, what we can do is we can run the same single command, but with just an extra argument to tell it how to run those models differently. And we run that, and it does the same thing, similar to before, except it spins up 100 VMs, runs each of the models, and outputs 100 maps of malaria risk. And so then what we can do with that is we can summarize those 100 maps to produce our predictions and with uncertainty. And so that takes exactly the same time as it would for one. And so this is where the power of Google Cloud comes in to help us. So if we compare this with our in-house on-premise servers, so I might have a single 64-core machine. So I've been talking about these global maps. We actually run the maps region by region, and we have 7 regions. So instead of just producing doing one run for the globe, we do 7 different. So on our on-premise servers, to produce 100 maps for a single region would take about a day at full capacity, and then we have to do it for each of the 7 regions, so producing global maps would take about a week. Whereas on Google Cloud, we can use many small machines and run each of the regions and the realizations, so these 100 maps, simultaneously. And that would then produce our outputs in less than a day. And we can also use preemptible machines that were mentioned earlier to reduce our costs. And so we're going from our turnaround time of producing global maps from about a week to less within the day. And this really benefits us because we work with organizations like the WHO, who are a big organization with many tight deadlines, quick turnaround times, and so it's important for us to be able to produce high-quality maps fast, basically. And so the less time it takes for us to run the models, the more time we can spend scrutinizing the results, iterating over the results, improving the model, improving our predictions, basically, for malaria. So here I'll just show some of our results. So this is malaria prevalence, so the proportion of people that have malaria globally from 2000 to 2017. So our models are vary in time as well. And you can see that the prevalence is much higher in sub-Saharan Africa. It's also going down with time. And so also what's important in these predictions is where is it increasing? We want to know that. We want to know, and why would it be increasing? Is there the lack of intervention? Is there development of insecticide resistance? So these maps can help us answer these kind of key questions in order to reduce malaria. Then we don't just produce prevalence, we also produce other things like mortality. So this is the number of deaths per 100,000 people each year. And so similarly, you can see it's high in sub-Saharan Africa, but this may be different to our prevalence estimates in the sense that you might just not get high prevalence, high mortality where you have high prevalence. It might also be affected by, do these people have access to healthcare? Do they have access to the effective treatments, things like this? And our group also works on modeling those and understanding those contributions to this stuff. So in summary, we produce global malaria risk maps using Pipelines API, and it makes the process about 10 times faster using Google Cloud. And this allows us to continually improve our models at a faster rate for the deadlines, and also it gives us scope to develop more complex and computationally intensive models going into the future. Thanks. Shall I move on? Is that the plan? Okay, so next I'll introduce Vivek from the Wellcome Sanger Institute.
Thank you. Brilliant. Thank you very much. Okay, hi everybody. I'm Vivek. I run the Human Genetics Informatics team at the Wellcome Sanger Institute, and I'll briefly go over the way that we've been using the GCP, and specifically Spark on the GCP, to run large-scale analysis of variation, human variation. I've got to say, I'm actually chuffed that I was actually asked to do this, and I'm very happy that I can actually share this story with you today. So thank you, Google. Okay. So what is Sanger? Sanger is a research institute in Cambridge, inside the UK. It sits in a genome campus. It's got about 1,000 staff. It sits on a campus which actually includes the European Bioinformatics Institute and various sort of scattered biotechs. It's a research institute based on a faculty model. Basically, it's subdivided into programs like HumGen, Pathogens, Cancer, and CellGen. And they rest atop a core of facilities, the sequencing and the IT. Specifically, I run a team that supports the Human Genetics program. And there are different research groups inside that program, for instance, that may research neurodevelopmental disorders, so that's a rare disease, IBD, which is a common disease, and actually complex trays, cardiometabolic trays, blood pressure, diabetes, this kind of thing. Okay. The broad aim of Sanger is to use genome sequences to advance the understanding of the biology of humans and pathogens and to improve human health. That's an extremely broad aim, right? But very specifically, the project I want to talk about is called Interval. It involves the whole genome sequencing of a cohort of 12,000 healthy individuals. These are walking and talking. They're not afflicted by any sort of nasty disease. Hamgen took those 12,000 individuals and the whole genome sequenced them at about 15x, a little bit more possibly, and ran bog standard GATK variant calling on them and got 185 million variants across 12,000 people. Okay. Critically to this cohort, though, not just the variants, but also that there are actually really thousands of measured phenotypic traits per person. So there are hundreds of blood cell counts, lipid levels, and whatnot. But also there's a full proteomic panel and metabolomics panel run on each person. Okay, and together they'll add up to a couple of thousand phenotypes. So those phenotypes actually give this study a quite unique window into the the molecular causes of complex disease, and it's a sort of a leg up on the average huge GWAS. Okay, so I've mentioned what the science is, but really this is sort of the packaging of the work. I've talked about the science, but really the thing I want to point out about the science is that it's actually urgent now. And the reason is that the landscape is competitive, and whilst this study was actually interesting and unique when it popped up 2 years ago, it's now getting steadily eclipsed by other large cohorts. Okay, so the field is competitive, especially if you think of UK Biobank. Okay. Specifically, then, the late delivery of this data means that it loses relevance. And whilst nobody's going to get fired or lose their jobs, we're all at risk of actually loss of future funding if this stuff actually shows up late. Okay. The work involved, as far as we are concerned, after the variant calling, which is sort of pretty standard, was, again, sort of standard variant QC. What makes it non-standard is that you're starting with a very large number of variants and a relatively large cohort. So when the QC runs, about 30% of those variants will actually be filtered out using standard sort of learned thresholds on various parameters. And then after that, the fun starts, because that's when all the association analyses start and the statistics gets going, which actually gets the association to the phenotypic trace. All right. The tool we decided to use for this was the HAIL framework by Broad. I like it. It's an expressive language. It's sort of basically like doing R. Okay, so it's actually critical for us to use this because it means that different people can actually look at each other's logic and very quickly understand whether that's actually appropriate or not. And there's a constant interaction between us and the postdocs and the faculty groups. So we have to be able to actually share the logic around and actually critique it. And if you code it up in a bundle of dense Python, that's actually hard to do. Secondly, where you're writing R-like stuff, it operates on a very large, high volumes of data. Okay, so it does things that R cannot, specifically because it's actually designed to run on top of an Apache Spark cluster. So that's actually why I chose that tool. I should point out again, before I leave this slide, that urgency is actually critical here. The landscape is competitive, and I can't afford delays of months while we sort out technical hoopla. Okay, so it turns out there's plenty of informatics support at Sanger, right? If you like, this is what our tech stack this is what the group stack looks like. We're in the middle, we're the Hamgen program, okay, but above us we are servicing the human genetics faculty, and we rely on the services of specialist high-performance specialists and on-prem cloud specialists below us, and they sort of rest on an IT core below them. So you might ask why on earth we're turning to GCP in the first place. And really, it's a combination of two things, right? So we're going sideways out to GCP for our Spark cluster services. So what's really happening is driven by urgency. That's the first thing. I can't afford to faff around waiting for my Spark clusters to come up locally. Second thing is, there's a strong precedent for Broad. So Broad wrote HAIL software, which is, of course, what we're using. They have a strong precedent for using Google to do all of their Spark processing. Okay, so given that, it's actually very easy to sort of follow down a well-worn path, and I'll explain why in a minute. In a sense, this is how the tech stack evolved. We started with traditional high-performance compute, which at Sanger we understand extremely well. Okay, we're really, really good at spraying stuff across large clusters and gathering up results. But I don't think that suits this particular task very well. We did and have built local Spark clusters. They are straightforward to build, as straightforward as anything else on an on-prem cloud. We found that the performance requires a lot of tuning. especially when you get to genome scale data sets, that's the first thing, and actually big cohorts. We've settled on using Spark clusters in Google Cloud in Dataproc. Why? Because it's easy to deploy. We have broad recipes that spin these things up in minutes. The provisioning is reliable. If it came up yesterday, it'll come up today. And the performance, this is critical, is actually predictable. Right? If it took an hour yesterday, $100 an hour, it'll do the same thing today. And we're not getting buffeted by other users competing for these resources. And finally, when we hit problems, and certainly we hit problems, they can actually be debugged with Broad. That's quite important, because they've hit the problems before. And we write to them, they tell us, oh, try this obscure parameter, and hey la, it actually works. So what I'm going to take you through in the last part of this talk is, in a sense, the little bit of the jitter and the data flow, and a couple of things that were cool, and a couple of things to watch out for. So yeah, as you can see, Stanger's on top, Google's on the bottom, and there's stuff sort of flying back and forth from left to right, from top to bottom. The first thing that had to happen is we had to get legal and ethical approval for our human data to enter the cloud. This is important, okay? We took a set of due diligence questions to Broad about security and privacy. They promptly shot back a bunch of comprehensive answers, and then eventually our legal and ethical folks went, yep, you can do this. I should point out that whilst Google was prompt, within a matter of days, answered, the process internally to get through legal and ethical was actually anything but prompt, and the timescales are kind of, yeah, it can take a while, it can take a really long while, you just have to be ready for that. Then the good bit started, right? We got the data into Google, we ran Spark and Hail to filter the variants. We took the filtered variants back out again, ran lots of plots to make sure the filtration was effective. When we realized it was effective, we ran Spark and Hail again to do the statistical analysis, and then took the results back out. And that all went swimmingly, right? It was reproducible, it was efficient, it was timely, everybody wins. This is a good thing. So GCP is doing really well. A few things you need to bear in mind when this happens. Firstly, we have tens of terabytes of data, which is not petabyte scales of stuff for a single project. Even so, storing hot data in the tens of terabyte scales inside Google will add up pretty fast when you have it there for months, for instance. That's one thing. Downloading terabytes of data will actually add up pretty fast as well. And of course, there's the processing time in Google. I mean, to do this effectively and quickly, we had to spin up hundreds of cores to actually get the job done. And that actually adds up as well. All of this can be managed, no problem. It can be predicted and managed. You just have to keep it in mind, and you have to be ready for that. Okay. It's all worth it, though, because the results come out in a way that we expect. Okay. And you can use those to write papers, which is what the game is. Which is the whole point, right? To actually sort of understand underlying biology of behind the variants, and to actually sort of eventually improve human health. Okay. I should say, and it's worth emphasizing why the timeliness of this whole thing is actually a big deal. Firstly, research groups have staff which turn over, often within 3 years, a typical postdoctoral fellow. It's not okay to sit on top of data for 6 months, because the postdoc might be leaving, okay? Or that's 6 months less that the postdoc gets to actually write the paper. It's totally not okay to actually sit on top of something for 6 months while your tech stack spins up. The second thing is, at the moment, this data set is the most deeply phenotyped data set, whole genome data set in the world. And it's actually effective for 2 reasons. Firstly, it can add value to bigger GWAS studies, because it actually can point to molecular mechanisms behind associations that other big GWAS studies have actually simply indicated, where they've indicated a link between a variant and a phenotypic trait. It can actually skewer the molecular mechanism. The second thing is that it can act as a pilot for those big GWAS data studies when they're actually pondering deep phenotyping. This is a relatively small study with very deep phenotyping. All the more reason that this data actually has to get out, and the science has to get out as fast as possible. Right? The longer we sit on top of that, the less relevant all of that becomes. So that's basically what happened. And I'm saying is that GCP and Google and Dataproc have actually facilitated the rapid scientific turnaround of this data, and actually have helped us actually do the science faster. So with that, that's pretty much all I wanted to say. I wanted to close with a few acknowledgements. Firstly, Hatem Nawar and Brian Gibson from Google Cloud have made this much easier. They've facilitated this at pretty much every step of the way. The scientists involved in the interval project are Nicole Soranzo, who's the PI, and her postdocs, Kaushik, Claudia, and Hannes. Our local on-site specialists are Pete Clapham and Tim Cutts and their teams, and of course Tim Paterba and the Hail team at Broad have helped us immensely along the way as well. Thank you. Okay. We can take questions for any of us, actually. Or not. It's late.
Nice talk, both of you. So my first question was to Vivek is, so the statistical framework and the variant calling framework that you use inside Hail,
Yeah.
these are already predefined?
Yeah, they've been written by the Broad Hail team. So the variant processing framework is pretty standard. It's just filtration with various different query predicates and so on. The statistics is actually pretty basic. You either run your bog standard linear regression, or you run SCAT, which is the frequency-weighted burden testing kind of thing. And that's about it. It's about as good as it gets right now.
Have you tried deep variant?
Deep variant. OK, so we're talking about two different things. We haven't tried deep variant. That's the first thing. But secondly, what we're doing here is actually downstream. If you like, it's the secondary, tertiary analysis downstream of all the basic variant calling, which is just absolutely bog standard GATK.
So you took the decision to using the GCP because the Broad Institute influenced you, or you did the comparative study with the other?
Oh gosh, I wish we had been that systematic. Actually, no. We took the decision to use the GCP because we could. We could afford the cost, and the Broad had actually done this before, and we could spin stuff up like a piece of cake. It was just easy. There's like a one-line recipe they give you, and boink, up come your Spark clusters. Okay. Not just that, because we actually had local Spark clusters, and they were not performant. All right? And honestly, I don't have the time to mess around trying to make them performant. When you've got a PI breathing down your neck going, where's my data, where's my data? Yeah, so that's basically what happened.
Thank you.
Yeah.
I was trying to follow up on the question. In your presentation, you said that you have to transfer tens of terabytes of data, and you say it's easy, doable, just have to be prepared for it.
Yeah.
Can you elaborate on that?
Well, OK. We pushed something of the order of 10 terabytes into GCP, which is easy, right? It's getting the stuff back out again, so going in is free. Getting it back out again costs 12p per gigabyte. OK. That's one thing. Storing it is 83 pence per terabyte day. So again, you just have to add this up across a month. And then finally, when we run clusters, we run at about $100 an hour. But we run big clusters. But our total end-to-end filtration and QC and statistical analysis takes about 24 hours. So it's OK. I mean, the point is we know that's what's going to happen. We're just ready for it. Does that kind of get to what you were asking for, or?
Yes, to one aspect. Another aspect is that how did you do it? You pushed this through the wires, or you shipped it physically?
We definitely did not ship it. We just used GSUtil copy one way or the other for our sync. Yeah.
Okay, thank you.
OK.
Uh-oh.
Shout.
Hello.
There we go.
So I'm wondering more on a human level. From your experience, do you foresee a future where, for instance, your institute would have no clusters and you would work only on Google Cloud Platform?
At the moment, I think actually having both is actually pretty good, because we can actually play locally and then execute stuff for real in production in the sky. Are you asking whether I can see a trajectory? At the moment, I'm quite happy to have both ecosystems in going. But maybe that's just my mindset right now. It's hard to change, that's all.
So there's like a clear advantage on your computing times and having up-to-date...
That's a super hard comparison to make, right? Because it is clear that we can run stuff faster in the sky than we can locally. It's just true, OK? But it's not clear that that will always be the case if we actually put lots and lots of effort to making stuff run locally more performant. The question is it worthwhile? And that's a bogglingly hard thing to actually get. What is true is that running it in the sky was actually completely worthwhile when you count the scientific value and actually just the sort of client service value of actually getting data to a scientist in a hurry. Okay, but that's really hard to quantify.
Yeah. Thank you.
Cool.
Great. Well, we'll do one more. We'll do one more, but then I think we have to. We're about out of time.
Wrap it up.
I wanted to ask this question, but I just wanted to give other. So my question is to Anita and Nandi. You mentioned you train multiple models or hundreds of models by varying different parameters. Are these sort of predictive models, machine learning models, or?
In the case I was talking about, these were not machine learning models. They were more Bayesian, like geospatial kind of modeling. But we also run machine learning models in different, but not specifically on our global ones. Yeah.
Great. All right. Thank you all so much for coming, and thank you all for coming to Google Cloud Next. Enjoy the rest of your day.